I'm working with geomicrobiology and microbial ecology from the University of Bergen, Norway. I also hold a adjunct professor position in the Arctic University Center in Svalbard, which is the northernmost university. And my rationale for more than 20 years I've been working in the Arctic, and my motivation to go into the Arctic was that I was studying soil microbiology and was so fascinated by this high diversity of soil microbes, and I also struggled so hard to understand the diversity and understand the mechanisms driving and establishing this diversity. So I figured out that if I went to the Arctic, it would probably be a very good laboratory to look into a very simple system and get to know this system much better. Because in the Arctic, as this image shows from Svalbard, you have very low diversity of higher organisms, plants and animals, and it looks very simple. So this was my motivation to go. But I've learned that this is not a very simple system. It is a lot of things buried in this system as it is. This IPCC report in 2018 was really kicking off the study and the research and study in Arctic ecosystems. And you probably all know this curve indicating that it's the CO2 emission is increasing, and this will increase the temperature on the globe, and the Arctic environment is really the one that is suffering the most. So we have already seen this image before, the permafrost, which is covering the Arctic, and 37% of the northern hemisphere is continuous permafrost. And then Svalbard is there where I have the adjunct position, and that's where I also do the most of my field work. So we have 60% of Svalbard is covered by glaciers, and we have a lot of areas with permafrost. I think this cartoon from the NASEM really shows a very good description of what's going on when you have a thawing permafrost. You have a regular, or the permafrost that it is, where it's quite distinct. We've seen quite a few images already with the ice wedges, and then you have little vegetation, and the carbon in and carbon out is really very much stable if you have a stable and not a thawing environment. When you have thawing of permafrost, you can see that the soil structure is decreasing. The plant starts growing faster. You have more biomass in the surface. You have more old carbon that is respired, and the ice wedges is melting. And then when you have, later on, you really see the collapse of the soil system, very little ice left, and now you see that we have a lot of carbon coming out of the soil system. I was saying that I've been working for 20 years in Svalbard, and I realized that it wasn't a very simple ecosystem to study, but I was very fascinated by studying it. And I have seen a very change in the system for the 20 years I've been there, and also very close to the settlement and where the buildings and everything is. So this is from Svalbard, summer 2017. This used to be a small lake in here. The lake is more or less gone, and then you can see a really rift in the soil system. So it was, and this is very close to settlements where the students are living, etc. So it's dramatic changes. If you go more into detail, you can really see the ice wedges here, and you can see the soil structure is exposed to the atmosphere. And here again is a closing up. And these ice wedges was only using a couple of months to thaw and to be gone. So then they whole and of course these are containing a lot of gases and a lot of carbon. So this is emitted also into the atmosphere. Okay. Astronomy has always amazed people for studying, and we actually know probably more about the galaxy and the stars up there than we actually know of our own planet. So this is not stars in the sky, but this is 1 gram of permafrost soil. which has been stained with a stain that binds to DNA. So this shows bacteria in a gram of permafrost soil. And there are 10 to the 10 bacteria in 1 gram of permafrost soil. And there are 10 to the 21 stars in the universe. And that's a huge number that we realize we can never really understand this in detail. And we have 10 to the 30 individual bacteria, microorganisms that we know of today. This is probably an underestimate. Most of these bacteria have never been cultivated and their functions are still unknown. And we know that these microbes are responsible for fundamental life processes on a global scale. And that this includes cycle of nitrogen and carbon and other nutrients. There are gigatons of carbon locked in the permafrost if the permafrost stays frozen. But if this is thawing, this means that we have induced microbial decomposition and releases of CO2 and methane in this system. The permafrost the carbon that are released from permafrost is 1.7 gigaton carbon per year. And that's very small compared to the anthropogenic release of carbon to the atmosphere. So still, it's less than the anthropogenic input, but we can't ignore these processes. Okay, so we have been studying. I mean, there is so much we don't know in the permafrost. I think some of my others on the panel will probably come back to more details later. But in order to understand this, what we're doing is that we're extracting the DNA from all these stars that you can see in the back there. We're trying to look into the genes, try to puzzle the genes together and see what functions are present in these systems. I have been working very much with permafrost from the Svalbard Longyearbyen. There is a distinct active layer. We have a transition zone around 1 meter, and then we have permafrost layer below 1 meter. You can see distinct different community profiles. It's very kind of known soil system in the active layer, and then you come down in the permafrost layer and you have a lot of gram-positive and spore-forming microbes in the system. But we also can see that the microbes are very important in these carbon processes. This is just because I don't have more time, I will not go into detail. I'm happy to discuss metagenomics and results from that, but this just shows one of the metagenomics DNA from 2 meters depth, showing that we had, just by looking into the genes, we could assemble 55 prokaryotic metagenome genomes from 13 different phyla. And then most of these were novel organisms that we've never seen before. We don't really know what they're doing. I'm super interested in this MAG, which is an iron oxidizer, because this is very different from any of the iron oxidizers that we've seen before. But this is the way we really are looking. So this is a big knowledge gap in the field, and we are really working hard to get further on this issue. What are just very brief results that we could see in this permafrost, what there was a lot of aerobic processes going on. We could see from the microbes living in the permafrost, it was a lot of stress response present, and we could also find really high abundance of antibiotic resistance genes in these old samples. So I think I'll stop with that.
I actually live in Arctic. I've been there for 10 years, so I live up there. And I think it's wonderful with all the research you all are doing, and all the knowledge is actually here in this room. That's amazing. On the other hand, I'm a doctor of veterinary medicine, so I do understand some of the science, but I also work for the government. That means I have to go back to my Inuit people and have to tell them, should we be scared now? What is under there? I also have to tell them what to do with all this knowledge, where to put it. And much more important, where are all that knowledge? Why is it not present in the Arctic? why it is so difficult for us to get the knowledge back home. So that's some of the questions I hope that you can help me solve. We hear about this wonderful system in the European Union with the E3, and we notify everything and we alert each other and blah, blah, blah. Wonderful. I live in Greenland. Luckily, I do have Denmark behind me, and they are very nice always to tell me if avian influenza is coming my way, or if there's some salmonella in a sausage which is sold in my shop, and things like that. So we are really thankful. But I really like to have, would be a wish for me, living in the Santa Claus country, of course, I would like to have somewhere where I can pick up the phone and say, can you help me? Where can I find knowledge? Is there some kind of database or whatever where we can log in as Inuit people and get the knowledge that you all have here now and all your colleagues around the world have? So that would be a wonderful thing. And it would be really, really nice to help to get some help to come back down into the action. What do we actually do? What kind of information do we give to ordinary people in ordinary lives? Because that's my dilemma. I work there with the ordinary people. Every day being a veterinarian, I talk with this Inuit hunter who has dogs. I talk to him about what his fears is, how he lives his life, how his life is changing because the ice is changing, because the hunting animals are changing their ways of living. It's in fact my life of living, even though I also live in a very postmodern society, which Greenland also is. So I would really, really hope that into tomorrow, maybe later today, we could also get a bit down in the action. What do we do? What do we tell ordinary people? So I'm so happy to be here. I feel a bit like the odd person. There's so many things I don't understand. So I'm happy that you do, and I really hope that somehow you can put this knowledge into my head, and that we can, thank you. Yeah.
While my slides are setting up, I would like to maybe respond to the comment about feeling like a fish out of water. I bet everyone here feels like that because it's such a broad group. So there's probably no one person who has a wrangle on all of it. So I think this is really important and interesting that we're all going to talk and figure out how our little teeny sub-disciplines all fit together for some greater good, right? So Lisa gave a really beautiful discussion on what microbes in permafrost are doing, that there's all this carbon in permafrost that's locked up, and that when permafrost is thawed, the microbes can decompose it, and talked about the importance of that because, of course, CO2 and methane are these really important greenhouse gases that we all care about in a warming world. And so I, along with some few other people actually on this panel, Robin and Tatiana, have been working to look at what the microbes in permafrost are, and we've been gathering, putting together a database to gather all the sequences that are known from permafrost. And so then we're hoping to try to work across studies to analyze what the microbes in the permafrost are doing when permafrost thaws. And so I will talk to you today a bit about what we're thinking about and how microbes in permafrost work, and then try to adapt it for what part pathogens might play there. So we're putting together a little paper right now, and this is a beautiful figure one of our colleagues made thinking about permafrost. So in the center we have undisturbed permafrost, and then we can go one of two directions, or maybe even along a continuum where we can have permafrost that becomes wet when it thaws, and permafrost that becomes dry when it thaws. And that will then affect which microorganisms are present and thrive. And so this figure, of course, is a little busy, but we are writing the paper thinking about carbon cycling. So we have things here like, we're going to have more methane production versus more CO2 production. These will be anaerobic processes versus aerobic when you go from wet to dry. And then we have some predictions based on the current literature about different types of microorganisms that will thrive and things like that. So some of this could potentially be important when we think about pathogens. We don't have pathogens explicitly on there at this point, and maybe we should as we put this together. So I kind of wanted to recap what is known about the permafrost microbiome at present, and these are just from what's out there in the current literature. So we know that the active layer and the permafrost microbial communities are not the same, that as you go down in layers, different things are happening. And as Lisa mentioned, a lot more is known about the active layer microbiome than the permafrost microbiome. We do know that the diversity of bacteria and fungi decreases with depth, and I think, and we'll analyze this in better shape when we get to putting the whole database together, but we think archaea increases in diversity as you move down the permafrost profile. We think that microbes live in brine channels in frozen permafrost. So, because as the soil freezes up, you have these really briny pockets that end up being able to host the life that's surviving in there. It seems from a few different people's work, and now I know I need to look at some of your work to add it to that list, that microbes are selected for long-term survival in permafrost. And so when I mean selected, we're talking about the adaptation and evolution of these microorganisms. It's particularly geared to actually survive these long-term conditions in the frozen soil. And that's different than a lot of soils where there is selection, but it's not going to be just selection based on survival, perhaps. And then we also know that when permafrost thaws, community composition shifts. And so when people take permafrost either into the lab, and we call them microcosms, but really what it is you just put permafrost into a jar and seal it up and put it in a fridge. But it sounds fancier to say it's microcosms and not canning jars of soil. But what we know from that is that we have this great diversity in permafrost that then changes when the permafrost thaws. So we know that these communities are different. We don't quite know if that's just because there was a lot of relic DNA in the frozen permafrost, and so these are some open questions still on the table. One of the things we're trying to do as we pull together all of these studies is to ask if we can predict how the microbial communities change, because that could be really helpful for us in order to predict greenhouse gases, but now also maybe pathogens, right? Or other things we need to know. So here's some of the things we don't know about the thawed permafrost microbiome. We don't actually know, and it feels silly to have to say this, but what proportion of the microbes survive the thaw of permafrost. Again, I said we look at it with the sequencing techniques, but those aren't quantitative. And so we need to be reassessing some of our tools with that. We don't know if there's a biological mechanism for whether or not a certain microorganism would survive the thaw or not, or if it's just a physical mechanism. Maybe it's close to a mineral surface that's somehow protecting it. Maybe it's completely random. And all of that has some implications for our ability to predict what happens with permafrost thaw. Given that permafrost microbes are selected for survival, can they compete with the newly dispersed active layer microbes? So we can expect, of course, that as permafrost thaws, and I think there have been so many nice pictures showing how the layers of soil change when permafrost thaws, how will those microbes from the active layer trickle down and compete? A few of us have done studies where you see that the microorganisms from the active layer actually seem that they have a lot more ability to degrade carbon much more rapidly than the permafrost microbes. So maybe they'll outcompete the permafrost microbes. And then let's see. So do increasing biotic interactions affect the community composition? That would be important as these brine channels are no longer there. Microbes are able to compete with each other and come into more interactions. And how do, of course, do these things play out over space and time? So I want to show this in a pictorial sense now. Oh, and by the way, that was in Abisko, Sweden. So this is an undergraduate, Katie Bennett from University of New Hampshire, up there doing methane measurements 2 summers ago. And this thermokarst, I guess it would be, Vlad will tell me I'm wrong. I always say it's a thermokarst, and then he'll be, I'm like, oh, good. Yay, I got a thumbs up. Developed actually in a very short amount of time, just a few years. And so, you know, you're noticing through the thread through these talks that some of these processes are just quite rapid. Okay, so here's the pictorial sense of why does community assembly matter in thawed permafrost? And I guess maybe I'll say the spoiler alert is that I think a lot of these things is we don't know, and then that knowing this, I think, will help us to better understand whether or not a certain member, if we find a pathogen in the soil, whether or not that actually would be able to grow to a virulent population. Okay, so here I have my beautiful drawing of a permafrost table as it goes from being intact to thawing, to whether or not this is space or time, but just how things are changing here. So this here now is the selective filter of permafrost. So we're going to have a permafrost community here at the top going through the conditions of living in permafrost. It's frozen all the time. It's very briny, just harsh conditions for the permafrost microbial community. And so that ends up with this community that's specially selected for life in permafrost. And that's been found by a few different studies. There's not a lot of dispersal from the active layer, we don't think, because of the frozen conditions, although there's some indication there could be some. Okay, so then when permafrost thaws, you actually have a change in the selective pressures. The change from frozen conditions to thawed conditions, that temperature transition above zero is very harsh for a living organism. So then you go from having this kind of already selected community to maybe just some other subset. And I know this is overly simplistic. It's not going to be one type of microbe, but I didn't want to show all the complexity, all the stars in space. You're also going to now have dispersal from the active layer straight down into that community, and they might not be selected to be there. So whether or not they're good at outcompeting the permafrost microorganisms or not, who knows? It's no longer a permafrost environment exactly, so we just don't quite know. So the community is now maybe being affected by some random losses in this transition, in this physical transition, and then also by dispersal, which makes it harder to predict what the things are going to do. If organisms are selected for their environment, it's very easy to look at their environment and predict what they're going to do. But if they're just randomly selected or randomly there, it's much harder to know what they're going to do. And then as things go through space and time, we might expect even to have further dispersal, maybe some speciation, who knows what we're going to have at that point. And so all of this to say, we need to know more about the permafrost microbiome in order to predict what would happen with the pathogens in it, or whether or not they can survive and thrive. So here are the questions I think that we would want to be able to answer out of thinking in this microbiome and well, we say we're studying the assembly of permafrost microbiomes, so what part the pathogens will play. So this, of course, would be assuming we would find the sequences or the microbes there, or the viruses, sorry. Are pathogens in permafrost active or dormant? If they're active in permafrost, maybe they're also selected for frozen conditions and won't survive the thaw. Who knows? If they're dormant, maybe they will. Will they be able to thrive and grow to a required population size, which I don't know if that's actually needed, so I would have to know that from the rest of the community, but to be an effective pathogen. There's going to be all these new temperature, substrate, moisture niches that are opening. Are they successful? Will pathogen populations be kept in check by the diversity and size of the community that's already there? That's somehow how we think things work in other soil systems.
How do you define pathogen in the system?
Well, I have to open that to the room. I mean, I think in my mind, I'm thinking about bacterial pathogens, but we do know that viruses regulate bacterial populations. There could be a fungal pathogen, a plant pathogen that would infect. If it infects a plant, that will change the community composition. So I don't know that I quite have the answer to that. I'm thinking I'm more in a hand wavy space. Sorry. Will the low biomass and diversity that I imagine might happen after the thaw allow pathogens to take a foothold in the community? And then is this even something that we could predict? I wrote here, is a pathogen dominance one of many alternate stable states? And I'm realizing that's probably like a way too ecology theoretical thing to say. What I mean by that is, if we thawed permafrost 1,000 times, what percentage of that would have a pathogen population that was large enough to be infectious? And would that be predictable, or is it just completely random? I think that's where I'm at. Oh, so just to let you know, where we're pulling data from all sorts of papers, trying to compile it, and that's kind of the current status. And so I've been taking screenshots of when people say this is a pathogen, I'm going to look through the sequence data and see if I can see any of it, I guess, at some point, but not by tomorrow, I apologize. But really, there aren't that many studies, so it's just a handful of studies we're looking at here. I think we have really 20 thaw experiments in our database. There's more of our active layer or permafrost, but in the database, that's it. So, yeah, thank you.
Thank you very much.
It was a nice introduction by Dr. Romanovsky about what permafrost is, and I'm not going to repeat him, but I would like to show this first slide where permafrost core is. That particular permafrost core we drilled from 50 meters deep in Alazea River site on Kaluma lowland, and that permafrost, as estimated, was formed between 1, 2, and 2 million years ago. So this is very old permafrost, and that old permafrost you can find just on Kaluma lowland. And that site is very interesting because of this very old permafrost. And collecting permafrost, it's very important to pay attention to aseptic techniques, how you collect permafrost and don't contaminate these unique samples with some bacteria from upper layer or from river air. Every bacteria you can find everywhere. And during 2 last field season in 2015 and 2017, we created short videos, and they are available on YouTube. You can just see where we worked and how we collected these permafrost samples. Actually, Kaluma lowland, this is a long, long way from University of Tennessee. And, oh, no, what I did. Where is it? Okay. And it was estimated that prokaryotic diversity in soil reaches between 6,000 to 38,000 species per gram. This is starry sky. And what part of that diversity we know? I think just little part. And this table I combined from different paper, and these studies showed a number of cells in 1 gram of permafrost counted by different techniques. This is total cell count, means that you stain cells in soil and count them under microscope. And you can see that it reaches from 5 degrees in Antarctic to 9 degrees in Siberia. Then you may calculate biomass from this actually knowledge. And viable count, it means that you just make soil suspension and then plate on Petri dish and count whatever group. And because you may use different media, you can get different microbial community on your Petri dishes. And microbial community you can grow, and this is just a small portion of that bacterial cells you count under microscope. Some of these community were studied, and this is just example of different Petri dishes And actually, this is different media, and you can see that media can influence community structure of that bacteria. Even this is grew from one soil. This is just media influence bacterial community. And this depends from researcher what media he uses. You can get different community structure. And again, this is a combined graph from different studies. And these studies describe viable, actually cultivable bacteria which grew on Petri dishes. Researchers use different media, but I did not put actually media composition, but idea that and the studies were devoted just like not more than 50, 60 bacterial strains per study. This is just small portion. But if you look on composition of these bacterial communities, you can see that Proteobacteria, Actinobacteria, Firmicutes, and Bacteroidetes group present pretty much. Bacteroides not, but Proteobacteria, Actinobacteria, and Firmicutes present in all studies. Then this is picture from different studies which were conducted in soil cryology lab in Pushchino. And actually, I worked in that lab before I moved to US, and I did my PhD study on diversity of microorganisms in columnar lowland permafrost. So what actually we found in permafrost? So we found different prokaryotic microorganisms, bacteria. We found archaea, found bacteria, found cyanobacteria, which were able to survive in permafrost. And that eukaryotic bacteria, again, we found fungi, yeast fungi, green algae. We were able to revive moss, even plant. Also found protista, and recent finding, this is nematode. So this was all these organisms were isolated from Siberian permafrost. Their samples were collected from different parts of columnar lowland. And this is actually from very old permafrost. Yes, and I did not mention it that viruses were isolated from some of these nematodes. But because cryology lab in Pushchino does work with viruses, so I did not put viruses into this picture, but viruses were also isolated, and we heard a nice presentation about viruses, old viruses today. And this table shows you type strains which were isolated from permafrost. Only 26 strains were identified and published. And this is, I put all these strains in this table. Mostly this is bacteria, methanogenic archaea, a few protista, and 2 giant viruses. And now to metagenome. And we have more and more metagenomes obtained from permafrost. And this is just data from metagenomes which are available on MGRAST. this is Metagenome database, publicly available, and I just downloaded metagenomes which I found in their databases. And I found 37 metagenomes from Permafrost, and if you calculate, if you do like plot microbial community, you from this all these 37 metagenomes, you can see that only only a small portion of these bacteria exist at level more than 1%, and exist at percentage more than 1%. And you see, this is all these bacteria comprise 94% of community. And what exactly these bacteria are? And I put stars where we found viable bacteria. So this is mostly bacteria. This is actinobacteria, protobacteria, firmicutes, bacteroidetes. We found we can isolate the bacteria on Petri dishes. Then cyanobacteria, we found them. Euarchaeota, mostly this is metanogens. We also found them. But a lot of community, this is just very small portion of soil community. You see, all this present in permafrost, and maybe they do something, and maybe they survive, but this is very difficult to find them. We need to use specific enrichment media to find some of these bacteria. And now I want to show you same for active layer metagenomes. So I found 26 metagenomes from active layer. If we make graph of bacterial community composition, this is graph pretty similar to permafrost, but you see we have protobacteria goes up, and actinobacteria just became lower amount in active layer. Because protobacteria contain a lot of bacteria which is doing something in nature. Methane oxidizers, or a lot of bacteria just belong to protobacteria. And all these functions goes in active layer. So maybe this is just community represent what community active layer and permafrost is different. And as Jessica mentioned it, that maybe permafrost, this is a sub-community, and some bacteria, if soil frozen, some bacteria just survive inside permafrost, but others just die. And if we, this is just summary table from previous two graphs, and this is, oh, sorry. Could you find me last? No. And this is just I put bacteria which represent which occur at level higher than 1%, and this level genus level. So all other bacteria, they are less than 1% in that metagenomes.
I think in the interest of time, Tatiana.
Okay, yes, and this is actually my last slide anyway. And I just want to thank my colleagues from US and Russia and NSF and Russian foundations for support this research.
Thank you.
My name's Dave Stanton. I'll try and keep this really brief because I know we're massively over on time. I thought this picture was quite relevant with Halloween being last week, but you'll be pleased to know it shouldn't directly do you any harm anyway, because this died quite a long time ago. Although I suppose whether it'll indirectly do you any harm is kind of the topic of this this workshop. So yeah, I'm based at the Swedish Museum of Natural History. I'm a Marie Curie Fellow, and I'm doing research on evolutionary genetics. As you can probably tell from that picture as well, I'm not a microbiologist. I work on larger animals, and we're interested in, well, me personally interested in carnivores mostly, and questions about wolf evolution and other sort of exotic species as well. But I would argue that in terms of the pathogens which might potentially infect us, studying those in some of these kind of animals perhaps could be quite illuminating, as you know, rather than the permafrost, I suppose they're more likely to be able to cross over to us. So yeah, our research lab works on a huge range of different species. So these are all these animals have all been found in permafrost, mostly from tunnels dug into the permafrost by Russian miners, and we work with a lot of Russian collaborators and Russian institutes to gain access to samples from these. This is the one from the first picture there, so it's about 30,000 years old. So we had a talk earlier today asking about the preservation of some of these animals, and you can get absolutely remarkable preservation. This is about, all these samples are over 15,000 years old, up to ages greater than radiocarbon dating. So we've got woolly rhino foot here, a mammoth. This is a Pleistocene bird, as far as we know, the oldest sort of intact remains of a bird ever found. So this is almost 40,000 years old, a cave lion, woolly rhino, wolf pup, and this looks like a lemming which has been stepped on by a mammoth or something. So yeah, but generally the approach that we use is we extract DNA out of these samples, we build genetic libraries and we sequence them, but we do sort of a shotgun sequencing approach, and what that means is quite often we have very low amounts of the actual species that we're interested in that, so it might be 1% of cave lion DNA. The rest to us is just kind of wasted data, and that is bacterial DNA, and other, well, you know, all these kind of potential pathogens that are contained in that data. So we're potentially gathering a huge amount of data that could be used to investigate some of these pathogens. And like I say, I'm not a microbiologist, but this sort of metabarcoding approach could be very valuable in our data. I did a quick, and it's something that's relatively straightforward to do as well. I mean, you just need some kind of reference database that you map to. And I just did, with some of my cave lion samples, I did some mapping for the anthrax genome. And I found that my samples, Boris and Spartak, these are two of the sort of soft tissue samples that we have, had quite a high proportion of anthrax reads in them. On closer inspection, so I was a little bit worried about that, but on closer inspection, you can see that these are sort of quite conserved regions, and so it's probably a relative of anthrax or something like that, but certainly nothing to worry about with our samples. But you know, like I say, this isn't something that I'm actively involved in, but something that could quite easily be done if the interest was there. And you know, investigate some other questions, whether these sort of results depend on the location that we find them, how old the samples are, etc., etc. But I mean, I think the one point I want to make quite clearly is that you know, even if we find these things, it doesn't necessarily mean that they're viable. And this is sort of some of the this is just looking at the This graph here shows the fragments of DNA that we have in our sequencing libraries, and they're all very fragmented, short pieces. And that's the thing, this DNA is very, very old, it's very damaged, and generally very, very small. So I mean, most of what we get is around 70 to 80 base pairs, and to get up to the anthrax genome, which I think is about 5 million base pairs, if this was sort of a real, this shows the prevalence and the size here. The anthrax genome would be sort of 2 miles up that way somewhere, and you can see it drops to 0 at around 1,000 base pairs. You know, it doesn't mean it's not there, but certainly most of this DNA is going to be quite fragmented. So yeah, we were working with collaboration with some Norwegians who did a similar kind of analysis in some of our mammoth samples. You know, the most common thing that they found was just a fairly innocuous bacterium that's found commonly in the soil. They did find some potentially toxic hits as well. So this particular Clostridium botulinum, yeah, it is very toxic and it leads to food poisoning. But yeah, I mean, I think unless you're going to be eating the samples, I don't think it's of any particular concern to us at the moment. So yeah, I mean, that's it really. But you know, I would say that I think there is a great potential with some of these samples, especially when we're, you know, it seems like a lot of the things that are missing in terms of information at the moment is sort of what is present, what are the potential risks, and that is something that we could sort of identify in our data, and it could also be a real good insight into sort of, you know, with these ancient samples, we could look at sort of the evolutionary history of some of these parasites as well, and I think that would be a really useful tool for a number of different applications, including developing treatments and vaccines and things like that if this does become an issue in the future. So thank you for listening.